Good afternoon, class. It's nice to be with you this semester, and with this time change on this past Sunday, we are able to see the sunlight, and I think today is a good day weather-wise, in contrast to weekend, the Saturday, Sunday. So today's or this week learning activities consists of learning about microbial technology. And following this, we will discuss this paper on Thursday. So today we talk about microbial biotechnology. And following this on Thursday, we talk about this paper on soil microbes. It's a very interesting topic. I will cover this on Thursday. Today, I believe, is Tuesday, and this is the assignment. Assignment has two parts, one from the slides and the other from the paper. Now, one thing I was very curious to ask this question in the discussion board with all of you is that, let me create this forum. Let's call it discussion board number 4. And let's give a timeline of, not 22, let's give a timeline of 28th by 11:59 p.m. The question that I want to ask you is that, what is the, or let's say, what are the differences and similarities between epidemic, pandemic, and endemic? That's a very general question. You can find this answer for this with the click of mouse on Google. But I want to know, what do you think? At what stage or spectrum we are at present moment with COVID-19, right? Meaning that, are we in epidemic phase or pandemic phase or endemic phase? Right? We are in some phase, right? We could be in between two phases, between, let's say, epidemic or pandemic or endemic, right? So what do you think? In what phase or phases we are at the present moment of COVID-19 with the spread of COVID-19, right? Nobody knows, but I want to know what you're thinking. So support your answers based on support your answers with a logical explanation. It's not a multiple choice question that you'll pick one. You have to explain what one you're choosing. Also, what are the differences between spread of HIV, or let's say what was the recent spread, Zika virus and COVID-19. So I don't know whether you remember or not, Zika also started on a very strong potential of pandemic or epidemic threat, but it was very quickly controlled by Obama administration. It's possible that the root of the spread, the disease may not be as severe as COVID-19 or coronavirus, but Zika virus did infect a certain subset of human population, particularly pregnant ladies, and also it emerged from South Africa. So I just want to know what are the difference between the spread of these two viruses. So I have given you date till today is 15th, and you have till until 28th. So let me increase this to 31st. So you almost have 2 weeks to answer this question on the discussion board, and write what comes in your mind, but do some homework, but support your answer in a logical way. So I hope time-wise I am reasonable for 2 weeks. Yeah. So you have the learning activities for this week is to lecture, slides. Next Thursday, coming Thursday, we talk about this paper. There will be few questions from this paper, not many, but at least we will go into one of the important aspect of soil microbiology or soil microbes, how they are important. And then we have the assignment. So let's go to the slides. Now, very common word, why is microbial biotechnology important? So one of the upfront reasons that I can simply say is that we have very limited antibiotics on the shelves right now. And population may not be in US, because in US the prescription of antibiotics is very controlled, but all over the world, and also when we get antibiotics, the physician emphasize and the pharmacist emphasize that we have to finish the course. Why we have to finish the course? Because it can give rise to drug resistant organisms, microorganisms. But all around the world, not all around the world, especially in people where healthcare is not very significantly emphasized by the government or healthcare policies, their use of antibiotic is very, very vague, and they don't know the concept of antibiotic resistance, or next time the antibiotics will not work if one is infected with the same organisms. So microbial technology, one of the aspects of microbial technology is to develop new therapeutic drugs that can be useful. For example, let's say septicemia, right? Septicemia happens in ICUs. So suddenly you will hear that a patient was doing fine but got infected in ICU. So ICU are the places where very robust multi-resistant bacteria grow in hospital settings. Understood, right? Because the environment is like that inside hospital ICU. So if there's somebody slightly immunocompromised, they have a high chances of getting antibiotics. They are given with multiple types of antibiotics, but they may not affect because we have very limited choice. So I have a collaborator at Rockefeller. So what he has developed a new potential candidate for these drug resistant bacteria, especially in ICU, especially in those patients with septicemia. So his research focus on that, let's say this is a bacteria which is drug resistant. This particular collaborator of mine, very smart strategy. He works with uh, bacteriophages, right? Now, this bacteriophages, they have capacity. These bacteriophages, they have the capacity to punch hole in the bacteria, right? They can punch hole in the bacteria. Yeah, so they can um, these bacteriophages, they can punch hole in the bacteria. And once inside the bacteria, these bacteriophages also releases a protein called as, this protein is called as lysines, right? These lysines, they can uh, break the bacterial cell wall. So this particular friend of mine, collaborator of mine, is working with lysine as a novel therapeutic target, therapeutic modality for for controlling the spread of drug-resistant bacteria, because they might be resistant to tetracycline, penicillin, clindamycin, right? So the new strategy could be a phage protein, because phages, they love bacteria. So if you get a protein that can break bacterial cell wall, we are in business. So that's a sort of a good example to explain the potential of microbial technology. So besides their antibiotic resistant, there are tons of things, right? For example, using bacteria, we can produce, I have mentioned this many times in my class. I have set the theme of my class using this example of, uh, insulin, that the recombinant insulin is produced inside bacterial body. So insulin is one of the recombinant product. Now there are many factors. All these interferons, they are immunomodulators, right? You might have heard during in very early COVID pandemic or epidemic. COVID spread during March of 2020 or April, very early, that some patients were given interferon as a treatment, because interferons have antiviral activity. Some patients were also given interleukin 2 to boost macrophage production, because it was being in early the concept was not clear. So many patients were given interleukin, which is type of immune modulator also, for boosting up immunity against to make the macrophage attack this COVID-19. So all these products are there, and which can be easily purchased if it is prescribed by the physician, depending upon the situation. Not all of this. I see, for example, beta galactosidase, that's for different thing. Follicle stimulating hormone, then tissue plasminogen activator to prevent clotting, or to control the blood clotting. Osteogenic protein in case some people have osteoporosis. Erythropoiesis stimulating protein in those patients who may have anemia, and so on and so forth. So bacteria serves as a body to produce these agents, which are proteins, recombinant proteins, and we have been discussing the strategy, restriction enzymes, vector, cut and paste, so on and so forth. So just to provide this information from a different angle, is that you take, so how do you, so let's say if I give you a project, right, a long answer question, and say that what is your favorite protein that you would like to open a biotechnology company. Some people can say insulin, some people can say, um, you know, hemoglobin to treat patients with sickle cell anemia. So you can come up with your own ideas, but to make a company, biotech company, which would be based on platform of recombinant DNA technology, you need a starting point. You need a human gene that would express insulin or hemoglobin or any other interferons, for your company. So what you do, from human leukocytes, you extract mRNA. This 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 mRNA should have the mRNA for, let's say, insulin, uh, and uh, and from there, from this mRNA through reverse transcription, you 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 get cDNA. cDNA is cloned into a plasmid. Plasmids. How did I identify this as a plasmid? Because plasmid is always written with a small p, not with a big P. So the cDNA for insulin is cloned into this plasmid, and then it is transformed into E. coli. Transformation of E. coli means that you shuttle the vector, the plasmid, recombinant plasmid, into the bacteria, right? And then you select those plasmid which are resistant for antibiotics. And then you grow these bacteria in a test tube and then produce the protein. And then here they have given the example of interferon. And then this interferon is extracted and produced. Now, one of the very popular microbial agent, a microbe, is yeast. Yeast is very much used to scale up the production of recombinant protein, because first it is an aerobic organism, eukaryotes, as contrast to bacteria, and you can control the temperature, you can grow it in large amount, and then again come back and uh, produce the protein. Yeast can be used for food culture, for production of food, beverages, so and so forth. And also it's used for research because it is an eukaryote, and it has all the not all the, but many similar transcription factors and signal pathways as a human cell. So yeast model serves as it may not replicate, it may not mimic human body, but it can serve as a model to spark some ideas to understand what goes on human body. And so a recombinant yeast is produced, again, through the same strategy: get the human gene and clone it in the plasmid that can be inserted into yeast. And then this yeast can be put into a fermenter tank, and by controlling the temperature and nourishment, a large amount of recombinant yeast is produced, and yeast secretes or has its recombinant protein, and that is derived through protein purification. And then same strategy of fermentation can be used for producing cheese, beverages, and different kinds of chocolates, so and so forth. And also the waste product of yeast can be, for yeast fermentation, could be used for agriculture or something else. Now, there are many vaccines that have been used, that have been produced using microbes, and they are very successful. For example, polio. We don't have, very rarely encounter polio, but I am saying that with a pinch of salt, because you don't know that, unfortunately, this polio virus can mutate, and polio could come back, because these viruses, they have tendency to mutate, right? And so that's the case with measles also. So recently, not recently, but several years back, there was a trial of measles vaccine in Europe, and the subject for trial, where the subjects who were involved in this trial were kids, and there were 100 kids enrolled into this clinical trial, and then out of 100, three kids, they got measles. So people were surprised that despite giving them, despite being vaccinated, how come they developed measles? So when they did the genotyping of these cases who became positive for measles, they found that there is an immune gene that is mutated, and that's why the measles virus measles virus could not be controlled by vaccines. So the idea of vaccine, we took vaccines, or those of us who took vaccine, we took with the notion that this vaccine will create memory within our body, such that when we encounter the variants of COVID-19, or any other vaccine that we get, when they counter the original bug, then we have the strong army in our body as a vaccine or immune system to fight, to counter those viruses. And there are there is chicken pox, smallpox vaccine, dengue virus vaccine, hepatitis vaccine, they're coming up, rotavirus. So I feel that vaccine is, in addition to medicines, drugs, molecules, vaccine is also important strategy. So vaccine is an agent that creates memory in our body, and that memory is kept in lymph nodes, mainly in immune cells. And whenever there is a fresh infection by the same bug or agent, microbial agent, these memory cells, they sprung into action and try to defend our body. So in what is simply, it can be, vaccines could be that the real pathogen is attenuated. Attenuation means this pathogen, the virulence is the capacity of a pathogen to cause disease. So when a pathogen is attenuated, means it loses its ability to cause disease. But this attenuated pathogen can be used as a molecule to develop vaccines. Also, it is very important to understand, to characterize this pathogen at a molecular biology level, and we exactly come to know which protein within this pathogen is causing disease. And that is called as antigenic determinant. So what is antigen? Antigen is anything that is foreign to our body's capacity of eliciting immune response. So an antigen could be a pollen, spring, allergic season, there'll be a lot of pollen floating in the environment, and we take it and we might have bronchitis or asthma-like situation. So why is that? Because it creates immune reaction in our lungs, in the alveoli, where gas exchange happens between carbon dioxide. So an antigen could be a foreign particle that is capable of eliciting immune response. But remember, there is a caveat here. The caveat is that not every foreign agents could elicit immune response. Some can evade the immune response, right? Immune response has to capture these agents. So there are some agents that can dodge. So not all foreign agents are antigenic. But anyway, once that antigen, the foreign object is characterized, then it can be used as a vaccine to inject into the human body. This vaccine creates memory cells. These memory cells create long-term immunity, such that when we encounter this active pathogen, not attenuated pathogen, but really the new pathogen, then when it goes inside our body, then these memory cells can become active. From 2, they become 2 millions, and they counter this. So once a protein has been characterized, that it can be used to counter the replication or propagation of a pathogen, be it bacteria or virus, then the first stage will be to give the vaccine to create memory cells. Then to make these memory cells more robust, booster is given, right? And then, so there are various stages. Stage one, deactivated virus. In our situation that we got for COVID is mRNA vaccine, but mRNA vaccine is the latest technology. Initially, inactivated or deactivated virus, attenuated virus is given, whether it can produce vaccine or not, whether it has capacity to generate immune response or not. Second stage, booster is given so that there is a fingerprint of memory cells in our body, right? So that's why the reason we get the booster. Developing vaccine. So again, this will help you with the term paper also. mRNA is a very, very, very interesting technology where scientists were able to develop COVID-19 vaccine within a year. But generally, if you go through the protein route, it might take 10 years. So first is to establish the proof of principle that a molecule could be immunogenic to have a vaccine-like property. A thorough research has to be done in the lab. And all the data that has been done on the research in the research on the lab side should be reproducible. And then this goes into preclinical development, right? Where it is tested on, let's say, on mice, in rabbits, or in monkeys. Then it goes to clinical trial where human phase 1, 2, 3 trials happen at FDA. FDA looks at the data, and then it goes to manufacturing where all these vaccines. So a typical protein vaccine takes 10 years, but mRNA just took 1 year because of technology and approach. And I suggested you to listen to that video on mRNA. So a vaccine, what is vaccine made up of? So let's say in COVID-19 situation, it will have mRNA. That mRNA should be in a solution, which is the buffer. This solution should be very much, comfortable. They should be very comfortable to our blood system because it should not create adverse reaction. Whatever adverse reaction should come from mRNA. Then to stabilize these mRNA into our intramuscular, because most of us we got through muscles, there are adjuvants given. This is especially in case of proteins. These adjuvants increase the potency of a vaccine to generate immune reaction. So these adjuvants, they mediate slow release of vaccine into our body and slow development of immunological memory cells. Right. Also, these vaccines might contain antibiotics, so that so let's say there's a vaccine that is being kept for 2 years. Who knows? Let's say the refrigeration is bad, then it can get contaminated with bacteria, and a contaminated vaccine given to a person, the person will suffer. So some of the vaccines could be a cocktail of some antibiotics, so that there is no growth. Right? If you leave milk for 2 days in your kitchen table, all the bacteria and fungus will grow. Same with bread, same with vaccine. So there are antibiotics there. Then stability, right? To increase the shelf life. So the more stable a product is, the more profit a company will get, and more reliable will be, on more therapeutic potential there will be. So a vaccine. So you must be knowing, if when I took COVID-19 vaccine, they came up with a tube, with a bottle, small bottle. In that bottle, there was a solution that they put it in a syringe. What this solution has? This solution has those products, those things that will stabilize the vaccine. Now, so I talked to you about attenuated vaccine, right? So, there are various kinds of vaccines. Live attenuated, they're live. This virus, these virions, they're live, but they're attenuated. Means they have lost their capacity to cause disease. Or this could be inactivated. let's say they could be inactivated by heat. So what we need to boost immunity is just the viral protein. So you can heat up this virus or put some solution in some solution due to which virus gets inactivated. So the virus is dead, not pathogenic, but it can generate immune response. So live attenuated, inactivated. Uh, re, reassortant means this has been modified. So these are whole virus. They could be live attenuated, it could be inactivated or reassortant. Then if you go into protein levels or molecular biology level, then you need to understand the protein composition, the amino acid composition, so that one can develop a recombinant protein. And you can see that in this graph that measles, which was very high from 50 to 68, due to this measles vaccines, it has come down to touch the, this is x-axis. So microbial microbes, they serve, not only they cause disease, but they serve as an important lab-like potential to produce various kinds of vaccines. And here also, this is an example of hepatitis B virus. So you know the antigen, the protein that can cause disease in liver, hepatitis, it causes disease in liver and it causes jaundice. And so if you want to get hepatitis, if you want to produce hepatitis B vaccine, then we need to know the protein that can cause disease and to develop a recombinant clone and then to develop hepatitis B vaccine. That's the route. Now, so when we, with the development of protein technologies, proteomic technologies, genomic technologies, we come to know which protein is critical for infection. In most of the viruses, the envelope protein. So what is a virus made up of generally? A virus is generally made up of, first of all, its envelope. And within the envelope, there is a capsid protein. And within the capsid protein, there is DNA or RNA. On this envelope protein, there can be receptors. These receptors contain the marker, the protein that can interact with human cells, right? And then once this virus and human cells, they interact with each other, then this virus goes inside the cells. So then the bottom line is that the knowledge of this viral receptor is very important, right? So there could be multiple virus receptors, right? For example, on HIV, there is GP1, GP1, GP120. This GP stands for glycoprotein 120. There are also receptors like co-receptors like GP41. So there could be various kinds of receptors, but we have to know which receptor is most critical for causing viral entry into, because if you know which receptor is which protein is important on the viral surface, on the envelope protein, it will be easy to design a vaccine or a molecule that will intercept virus entry into human cells. So we have to identify which receptor is important. And receptors are generally proteinaceous in nature or glycoprotein. There could be lipid molecule also. So once we have the understanding of a protein, let's say this is protein on the surface of a virus, which is important, which helps the virus to enter into the cells. Then we try to break this protein and really understand which sequence of amino acids are critical. Then this potential peptide, which helps the virus to enter into the cells, antibodies could be developed so that this virus and human cell interaction is inhibited. And then the idea is to develop cellular immunity. Cellular immunity could have two branches. One is through B cells, the other should be from T cells. But this is not an immunology class, so I will not. Then once we know which receptor is important, then it is important to narrow down to the protein. For example, humans herpes simplex virus, right? The GD protein is important. Herpes simplex GD protein is important for entry of this virus into our human cells. Then this GD gene, the gene that makes GD protein, is cloned, right? And to test whether this GD protein could be useful, this GD protein is produced in CHO cell line. This is a hamster derived cell lines. And this GD protein is extracted, and two batches of mice are created. One has injected with GD protein from CHO cells, from recombinant GD protein. So this mice is pre-injected, and this mice is not injected. To test whether GD protein is effective or not, both the mice, the control and experimental, they are infected with the virus. So the one that does not receive the vaccine, GD protein, so GD protein is a vaccine candidate. So this batch of mice did not receive this candidate vaccine. So it was not protected. This was protected. So once we figure out the GD protein, we have to narrow down to amino acids, right? So if this is the GD protein, what is a GD protein? It is extracellular. It means that if this is a viral surface, viral membrane, it protrudes outside in the environment. It has a transmembrane that goes through the membrane, and then it is inside the virus body, right? So from this such a long GD protein, we have to narrow down to few amino acids which are important for facilitating entry. So this is the model for GD protein, right? And then through experimentation from whole organism to whole protein, then only so this GD protein on the extracellular side of virus, you have one domain, two domain, three domains, fourth domain, fifth domain. Out of this 5 extracellular domain envelope protein, GD protein, which domain is important for helping the virus to go inside, right? So that is found out. So it was found out, let's say that domain 2 or domain 3 is important. But these domains are very small, right? Very small. For example, it could be from amino acid 1 to 20, 21 to 40, 141 amino acid to 160. So while doing this experiment, scientists found that 141 to 160 peptide is important for infection. But remember, while doing science, there are advantages and disadvantages going on at the same time. But it depends what is more dominant and what are the priorities. So while you narrow down to peptide for vaccines, peptides are not effective vaccines. So they have to mix with a carrier protein to increase the immunogenicity. So I repeat, short peptides, 20 amino acid peptide, they're not immunogenic molecules. They cannot generate immunogenicity. They're too small. So they are conjugated with a ligand or adjuvant called as keyhole limpet hemocyanin. So this is a carrier protein, KLH. KLH. And when these peptides, short peptides, they are linked to this carrier peptide KLH, and this becomes very immunogenic. So there are various strategies that are used for vaccine development. Uh, Malaria disease, right? Tropical climate. There are countries where malaria is still very prevalent. It comes seasonal. It is very seasonal, and when it comes, it creates a lot of devastation. And why malaria is important? Because there are many parasitic. So malaria is caused by Plasmodium, uh, Plasmodium falciparum. I repeat, malaria is a disease that is caused by a parasite called as Plasmodium falciparum. This enters into our body through a mosquito, Anopheles mosquito. These mosquito, Anopheles mosquito, also can spread dengue and and Zika also, right? Same mosquito. So there must be some proteins inside the saliva of these mosquitoes, in which these parasite or viruses are comfortable. And once this mosquito sits on human skin, they can transmit this parasite or malaria, parasite malaria or viruses, Zika or dengue. Right? In tropical countries, these are very common. And people don't know whether it is malaria or virus, Zika, or dengue or some other virus. Now, the complexity with malarial parasite is that it can change shape. It could be a different parasite inside the blood. It could be a different parasite inside the liver cell. It could be a different parasite inside in the mouth of the. So it has two biology associated with. So the life cycle of a malarial parasite happens in two hosts. One is in mosquito, and the second is in human body. I don't want to go into the life cycle detail, but each phases produces a unique protein because of which the parasite can dodge human cells, human immune cells. So they mostly sit in liver, and now the recent, and so DDT was used. DDT is a chemical compound that was used, very forcefully across developing countries to control malarial spread through the mosquito. So DDT would target the mosquito. So they would spread into water, river, ponds, stationary water. Then suddenly what happened, these mosquitoes became resistant to DDT and they had malaria that is resistant to DDT. So another level of problem. Then what happened that these parasites now can cross human blood-brain barrier. It can enter into the brain. Tuberculosis, which is supposed to be the disease of lungs, they can also cross brain. So cerebral malaria, cerebral tuberculosis. So the more we use medicines, antibiotics, without any care, the chances of drug-resistant parasite, bacteria, to occur in environment is very fast. And since human population is now very much globalized, people travel a lot. So the spread could be very fast. So vaccine becomes a very unique angle to treat these diseases. So, um, so we are doing a term paper on mRNA vaccines. We just talked about protein-based vaccines, right? Now there are DNA-based vaccine also, where DNA can be injected directly. The way mRNA was injected, DNA could be integrated into, I repeat, like mRNA, DNA could also be injected into our muscles, right? But DNA poses a problem that there are some protein that can help the plasmid to integrate into our chromosome. So if a bacterial plasmid or a viral plasmid which goes into our body with the intention that there is vaccine molecule, it gets integrated into our DNA, our chromosome, then it leads to another level of problem. So this is one of the advantage with mRNA vaccine is that mRNA cannot integrate into our DNA. mRNA just has to cross the cell membrane, go into the cytoplasm, meet a ribosome and make the protein. DNA has to enter into our cells through membrane, then through nuclear membrane, enter into the nuclear pool. In the nuclear pool, it has to undergo transcription, then again come back to cytoplasm. So there are multi-steps that are involved with DNA vaccines. And plus, toxicity is that it could integrate into our host DNA, into our chromosome. And let's say it gets integrated to a cancer genes, then it can easily cause cancers. So, I would stop here because I thought I gave a lot of information to you. In the next class, I will talk about, that's on Thursday, about that paper and about vitamin C probiotics, which is very important these days. A lot of probiotics are there on the pharmacy, inside the pharmacy, and they promises to uplift the digestion and so on and so forth. So probiotics and microbiome. So 4 topics, 4 subtopics we will discuss on Thursday, and it will finish this chapter. So I want you to ask questions now.
Good evening, Professor.
Good evening.
Sorry for the background noise.
Okay. You want to say something?
I came in a little late today, but I don't know if you took me for attendance. I should
May,
tell
may I know your name? Oh, okay.
Yeah.
Yeah, I saw you.
Okay, thank you.
Any questions? So if there are no questions, may I have the permission to end the class?
Okay.
And see you Thursday, same time.
Thank you, Professor.
I have a question.
Good evening.
I have a question.
All right, have a good evening.
Okay. Yeah, please go ahead with the question.
Oh, you're
No, no, sorry, no, not you.
Oh, I thought you were talking about too. I'm sorry.
You're fine, thank you. Hello?
Yes, hi.
Hi, I just wanted to make sure you have me down for attendance.
May I know your name?
Kayla?
Who? Oh, Kayla. Okay.
Kayla?
Okay, got you.
Okay, yeah. I was here in the beginning of class, but
Yeah,
okay.
I saw you.
You
Yeah.
got me. Okay, thank you.
So there are no questions, and I'm ending this meeting.